Hello and welcome to the post-lecture on glycolysis. I wanted to finish up the last handful of slides, picking up from where I left off at the end of class, talking about the Cori cycle and the relationship between glycolysis that happens under anaerobic conditions in skeletal muscle and the transport of lactate into the liver, where gluconeogenesis is performed to convert that lactate back to pyruvate, and then pyruvate all the way up to glucose through an ATP-intensive process. Gluconeogenesis and glycolysis have a lot of steps in common. If you look at the thermodynamics of glycolysis overall, there are really only three irreversible steps that I want you to be aware of, because the irreversibility of those steps relates intimately to how those steps are reconfigured during gluconeogenesis. All of the other steps outside of those irreversible steps can go in the forward direction or the reverse direction, depending upon the concentration of products and reactants. They're that close from a delta G perspective that from a concentration-wise they can move forward or backwards. Those irreversible steps in glycolysis occur at the very beginning and at the very end. The very beginning irreversible steps are the phosphorylation of glucose, the first phosphorylation that's catalyzed by the enzyme hexokinase, and then the second phosphorylation catalyzed by phosphofructokinase. Each one of these, when the phosphate gets added onto the metabolite, it moves that metabolite into a higher energy state, but not such a high energy state that the phosphate could get transferred back to ATP. Instead, what occurs is that there are dedicated phosphatases that remove the inorganic phosphate to take that metabolite back to the precursor state in the glycolytic pathway. The third irreversible step is the last substrate level phosphorylation involving a kinase called pyruvate kinase. To remind, what's occurring at this step is that a phosphate from phosphoenolpyruvate gets transferred from onto ADP to make ATP and generate pyruvate. Here, the substrate, by virtue of that substrate level phosphorylation, is moved to such a low energy state that it's effectively impossible to have a high enough level of pyruvate for it to go in the reverse direction. So for gluconeogenesis to get back up to phosphoenolpyruvate, it uses another intermediate called oxaloacetate, which you'll hear more about in the next lecture, as a halfway point to get back to phosphoenolpyruvate, and then everything else can run through the reversible reactions of both glycolysis and gluconeogenesis. Gluconeogenesis occurs in the liver, but it also can occur in other circumstances when glucose is in short supply and there's a need to mobilize it. What happens if a cell has too much glucose, more than its energetic needs at the time? Well, in places like the muscle and also in the liver, excess glucose is stored in a polymerized form called glycogen. You can think of this as a safekeeping of the metabolic precursors that can be mobilized very rapidly, much more rapidly than if it had to come from another organ source or be mobilized by breakdown of other more complex processes, ways of generating ATP. Let's talk about how glycogen is made. The input into glycogen synthesis is that first phosphorylated form of glucose, let me go back one, glucose 6-phosphate here, catalyzed by hexokinase when it goes into glycogen synthesis. There is a mutase called phosphoglucomutase that moves that phosphate from the 6th position on the glucose to the 1 position. And then what occurs is that the glucose is conjugated to with a UTP, so a uridine triphosphate, releasing an inorganic pyrophosphate and generating what's called UDP glucose. So a diphosphate linked to uridine and the glucose molecule. And this UDP glucose acts as the monomer for a polymerization reaction that's catalyzed by an enzyme called glycogen synthase. The activity of glycogen synthase is regulated by phosphorylation, and it is regulated by things such as the insulin is available in the cell and the availability of glucose. When there's excess glucose 6-phosphate, there will be excess UDP glucose as a monomer that can get conjugated to this elongating glycogen chain as follows. And these glycogen chains are built up by dozens of glucose molecules at a time as the so-called 1,4 linkages, but then also 1,6 linkages to create branch forms that together can bring together up to 50,000 glucose molecules, monomers, as one glycogen polymer. With these glycogen molecules, they can then be utilized by running the reaction in a reverse. Let's say now we're starting with a large glycogen polymer as here, and we want to be able to mobilize that glucose for glycolysis. Well, what occurs is that there's another enzyme called glycogen phosphorylase that transfers an inorganic phosphate, and it's the transfer of this inorganic phosphate to glycogen which makes it a phosphorylase as opposed to a kinase, which would use ATP. By transferring that inorganic phosphate, it then releases a glucose 1-phosphate off the end of the glycogen polymer, and then that glucose 1-phosphate can be converted by a mutase back to glucose 6-phosphate, which, to remind, is the step 2 of glycolysis downstream of hexakinase. So to review, going into glycogen involves a glucose 6-phosphate to 1-phosphate, UDP glucose, and to glycogen. In the reverse, you simply go from glycogen to glucose 1-phosphate, back to glucose 6-phosphate, and into glycolysis from there. The relationship between glycolysis and gluconeogenesis is further related to the way in which glycolysis itself is controlled metabolically according to its intermediates and its enzyme activities. I mentioned those irreversible steps at hexakinase, phosphofructokinase, and pyruvate kinase, always comparing glycogen and gluconeogenesis. Those same three enzymes are subject to feedback control at feedback and feed-forward control at different points along the metabolic cascade. Because they're irreversible, those are the key control points for these metabolites to relate to. How are they regulated? So hexakinase is inhibited by its product, glucose 6-phosphate, in a form of negative feedback. So if there's lots of glucose, and then by extension lots of glucose 6-phosphate, excess glucose 6-phosphate is going to feed back negatively and inhibit the activity of hexakinase to generate less glucose 6-phosphate. There's a similar type of negative feedback inhibition at the level of phosphofructokinase, but its inhibition is not by its product, but by one of its downstream outputs of the pathway, namely ATP. If there are high levels of ATP in the cell, phosphofructokinase is inhibited, and then this acts as a regulatory point in the cells to keep them backed up at fructose 6-phosphate. This could run in reverse and be stored as glycogen molecules through glucose 6-phosphate. And then the final regulation is not a feedback, but a feed forward, impinging on pyruvate kinase at that final substrate level phosphorylation. The metabolic intermediate fructose 1,6-bisphosphate, which is the product of the phosphofructokinase catalyzed reaction, it activates pyruvate kinase. And the thinking here is that what's occurring is almost as an anticipation for more substrate level phosphorylation going to occur as that fructose 1,6-bisphosphate gets dehydrogenated and isomerized and moved down along the cascade. And so if there are high levels of fructose 1,6-bisphosphate, the enzyme's at maximal activity to prevent metabolic intermediates from backing up in these downstream pathways of glycolysis. Here, there aren't real exit points for those metabolic intermediates, and so at that point, it is most efficient for those products to go all the way through to completion and the formation of pyruvate. And those are the final handful of slides that I wanted to get through. As I mentioned, we'll cover through glycolysis and the Cori cycle on the quiz. The relationships between glycolysis and gluconeogenesis, glycogen storage, and utilization will come back on the very last quiz and the fair game for the final exam. I'll see you in class on Thursday.